Okay, good morning, everyone. In a last lecture, we discussed about cell cycle, few points which are related with the cell cycle we have to complete, and then we will switch over to the next topic, that is cell division, particularly mitosis. Now, in this particular topic related with the cell cycle, as you know, G1, S, G2, and M. These are the 4 subphases in a cell cycle. Particularly, G1 as G2 together constitute what is called as interphase, and remaining one is mitotic phase. Together it makes what is called as cell cycle. Now, what is the status of the chromosomes in case of a different phases of cell cycle? We will show you the status of a single chromosome, what it goes through. This would be applicable to all chromosomes of the cell. For example, if it is the human cell, same event will be applicable to all remaining 46 chromosomes. In a normal condition, our 2n number, our diploid chromosome number is 46. So here we are just looking at the fate of one chromosome out of that 46. Same thing happens to all 46 of them, or 23 pairs of them. So please understand. Let us assume that a cell has started its life entering into G1 stage. This is the cell I am talking about. And here is its one chromosome. As you know that before replication, chromosomes are single chromatid structures. After replication, they become two chromatid containing chromosome, the two sister chromatids containing chromosome, held together by the centromere. Already you have learned in chapter number 8, so I'm not going in detail. Now this cell starts its life in a G1 stage, and at some point it will enter in the S stage. S stage is the one in which, if this is the single chromatid containing chromosome, it will replicate itself. So this chromatid is getting duplicated. An identical copy is created over there. At this point, it is called as sister chromatid, held together by this centromere. So beginning of S phase, the process starts. By the end of S phase, the process is accomplished. So a chromosome which will look like this in the beginning, the same chromosome at the end will look like this in the form of duplicated one. So that is what happens with the chromosome. Now same shall carry on in the G2 phase. As it is, it will remain. And now while passing through the mitotic phase, in mitosis, each of this chromosome with the two sister chromatids, its two chromatids get separated. One chromatid goes to one daughter cell, another chromatid goes to another daughter cell. This happened with a one chromosome. So again, each daughter cell is starting its life with a chromosome having single chromatid. If this is the one, such 46 chromosomes available, so all 46 will become like this. And all 46 will divide like this. So duplication, division, duplication, division, in between same. Single chromatid chromosome with which the cell enters into G1 stage. Then in S stage, each chromosome with a single chromatid duplicates itself. So shall be having now two sister chromatids, each one such 46. If it is a human cell, 46 chromosomes are ready, each with two chromatids held by centromere. Same will be retained in the G2. And in M phase, mitotic phase, each of this chromosome will divide into two separate chromatids. So 46 chromosomes, each with two chromatids, will give rise to 46 single chromatids on one side and 46 single chromatids on other side. Each set will be there in the newly formed daughter cell. So that is what happens with chromosomes. At this stage, if somebody wants to understand the amount aspect of the chromosome, so अगर ये सेल में 2C था, तो इसके बिगिनिंग तक 2C रहता है, S के बिगिनिंग तक, S के एंड में ये अमाउंट क्या हो जाता है 4C. यहाँ पे भी क्या रहता है 4C, और यहाँ पे वापस 4C से स्टार्ट होता है और फिर 2C में डिवाइड हो जाता है, like that. So that is about the amount aspect. If somebody wants to understand the number of chromosomes, so cell always starts with 2n number of chromosomes. Beginning of S, it will be 2n. Actually, at this point, it is 2n plus 2n, or else you may even call it as a 4n. Same will remain in this one, 4n. Same is here in the beginning of M phase. And here it becomes again how much? 2n, 2n. So I hope you have understood the same. If you have any doubt, you ask. If you have no doubts, then I will go to the next one. If you do not understand this, you cannot understand mitosis and meiosis. I repeat, if you do not understand this, you cannot understand mitosis and meiosis. Things will never be clear to you. The chromosome number related questions as and when asked, chromatid number related questions as and when asked, you will never be able to reply correctly if you have not understood the same. If anyone having any doubt, ask. Otherwise, I go to the next topic. Well, no question being raised. That means you have no doubts. That means we can move to the next topic.
हेलो सर.
हाँ, कौन है भाई?
सर गाना.
हाँ, बोलिए.
સર જે 2C જ્યારે હોય ક્રોમોઝોમ ત્યારે 23 ક્રોમોઝોમ્સ હોય એની અંદર, અને 4C થાય ત્યારે 46 ક્રોમોઝોમ્સ થઈ જાય.
પહેલા જ 46 સિંગલ ક્રોમેટિડ વાળા હોય જીવનની બિગિનિંગમાં. Beginning of G1 stage itself, there will be 46 chromatids. By the end of S, there will be 46 chromosomes, each with two chromatids.
અચ્છા, ઓકે, હા, ઓકે.
Yeah.
Thank you, sir.
Yeah. So now we have
Hello,
two different
sir.
types of. हाँ बोलिए कौन?
Sir, Shreya.
Hmm.
Sir, so each chromatid will undergo this kind of cycle.
Yes,
Okay, sir.
we have 46 chromatids in our daughter cells. They all will become double chromatid structures at the end of S. Continue as it is in the G2, and they all will divide into again 46 chromatids in one cell and 46 chromatids in other cell. That's how it happens with the human cell. So there are two types of cells, two types of divisions. One is mitosis, another one is meiosis. We have to study two types of cell divisions: mitotic cell division, meiotic cell division. Let us begin with first mitosis, the brief introduction. As you can see that a diploid cell can go through mitosis and can make two diploid daughter cells. Or else it's a haploid cell which can also go through mitosis and can give rise to two daughter cells. Both are haploid. A diploid cell can go through mitosis. A haploid cell can also go through mitosis. So if somebody asks which type of cells can perform mitosis, your answer will be diploid cells as well as haploid cells. Both are capable of carrying out mitosis. On the other hand, you have a meiosis. Meiosis is the one in which back to back two rounds of division, in which a mother cell gives rise to two daughter cells, and each daughter cell further gives rise to two more daughter cells. So actually, from one mother cell, you get four daughter cells. And another aspect, it's only diploid cell which can divide to give rise to meiosis, to carry out the meiosis rather. Mitosis में क्या होता है? Chromosome numbers are maintained same as whatever is there in the mother cell, same is maintained in the daughter cell. अगर diploid cell divide हो रहा है तो अगर mother cell is diploid, then the daughter cells are also diploid. If a mother cell is haploid, then the daughter cells are also haploid. So that is the mitotic cell division. Look at the meiotic cell division. If the mother cell is diploid, the ultimate daughter cells that are formed, they are haploid. So actually chromosome number is reduced to half in each daughter cell. To do so, you require a diploid cell. Diploid का half करोगे तो haploid होगा ना. अगर haploid रखोगे to start with as a mother cell entering in the meiosis, how can you divide it and give rise to n by 2 in the daughter cell, which is technically not possible. n by 2 जैसी कोई phenomenon नहीं हो सकता है. एक set होता है उसका आधा set, it doesn't work. So either one set or two sets. That's the way the system is retaining the chromosomes. Diploid cells will have two sets of chromosomes. Haploid cells will have a single set of chromosomes. So all this understanding you must have. At this point, I have in the beginning of this mitotic and meiotic study, at an introductory level, I have explained to you 2 points of differences between mitosis and meiosis. Number one, In which cells the mitosis can be performed? A diploid cell as well as a haploid cell. Both can perform mitosis. In which cells meiosis can be performed? Only in diploid cells meiosis can be performed. Second difference. How many daughter cells are born at the end of mitosis? 2. How many daughter cells are born at the end of meiosis? 4. Third, what is the status of chromosome number at the end of mitosis? Same as is in mother cell. अगर डिप्लॉइड सेल डिवाइड होता है तो डॉटर सेल्स में 2 and 2 है, हैप्लॉइड सेल डिवाइड होता है तो डॉटर सेल्स में n है. तो व्हाट अबाउट मियोसिस? तीसरा डिफरेंस मैं बोल रहा हूं. The daughter cells possess half the number of chromosomes compared with the mother cell chromosome number. Now these are the 3 points of difference which you must have to understand in the beginning itself before we go in details of mitosis and later the details of meiosis. If this is clear, then I'll switch over to straight away mitosis. Which cells can perform mitosis? Chromosome number point of view, which cells can perform mitosis? Haploid as well as diploid cells both have a power to undergo mitosis. Which cells can perform meiosis? Only diploid. That's the better term. Diploid is wrong. Only diploid. How many daughter cells are formed at the end of mitosis? Whenever cell division occurs, mitotic division, how many cells are to be born? 2. What about if meiosis occurs? 4 daughter cells, because meiosis is not a one division. It's a set of 2 back-to-back divisions. So from one cell, 2 daughter cells, and each daughter cell will give rise to 2 more daughter cells. So literally from one cell you receive 4 daughter cells. Now, starting with the mitosis. Typically diploid cells go for mitosis, so we are going to talk about that form of mitosis, which is a very common form of mitosis. A mother cell, which is diploid, through this division gives rise to 2 daughter cells. Both are diploid over there. That's a typical representation of mitosis. This gives you a wrong impression. This kind of representation of mitosis gives you a wrong impression. You have to understand it in this manner, that there is a cell cycle in which there is a G1 phase, there is a S phase, there is G2 phase, and then there is M phase. The chromosome number in the G1 stage is 2n. I told you that every cell has to begin with 2n number of chromosome in its life, and beginning means G1 stage. Then in case of its S phase, each of this chromosome duplicates itself. It will duplicate itself. So you have 46 chromosomes only, but each with the 2 sister chromatids. So 2n plus 2n will make it 4n. Same status shall be maintained in G2, no change over there. And during the mitosis, one chromatid from this, each chromosome wait, just a minute, huh? Now, as you can see that this is the mother cell and this is the daughter cell. If this mother cell is 2n, so these daughter cells are also 2n. So they are showing us this picture of mitosis. Have a look. Do you understand what I am explaining? This is not the real picture of mitosis. This is the real picture of cell cycle. आप जिसको मदर सेल बोलते हो वो मदर सेल ये नहीं है कि जो एक्चुअली डिवीजन में एंटर होता है. आप इसको मदर सेल बोलते हो. दिस इज द मदर सेल. The one which entered in its cell cycle through its G1 stage is the real mother cell, which passes through its G1, which passes through S. During S, it duplicates its each chromatid. Same shall be maintained when it is passing through the G2. And then it enters in the mitosis, and passing through the mitosis, karyokinesis and cytokinesis. There it will give rise to 2 daughter cells where each one will be having back to its normal original number of chromosome 2n 2n. This is not the cell which enters into the mitosis. This is the cell which is actually entering into in its G1 stage of the cell cycle. That is what you need to understand. How many have understood that? Now, if you have any question, you ask. Otherwise, I'll go to the next one. Now, literally, इसको number 1 देता हूँ, इसको number oh, इसको number 1 दूं, इसको number 2 दूं, तो ऐसे 46 होते हैं. फिर इसको number 1 दूं, इसको number 2, तो ऐसे ही 46 chromosomes होते हैं. So that is the way which it happens in our normal human cell. If you understand this, it's great. If you don't understand, please ask me. I will give clarity on this one again if required, because without this you cannot go in details of mitosis. Really no problem. That is amazing. Again, I would say that generally we represent the mitosis in this manner, showing that a diploid mother cell giving rise to 2 daughter cells which are also diploid. Now, mathematically, this is not possible that you divide 2n by 2 and again you get 2n 2n. Is it possible? Is it possible?
No, sir.
No. तो क्या होना चाहिए? ऐसा होना चाहिए. 2n at some stage becomes 4n. That is in S stage. And then divide that 4n by 2. And then you can see, you can receive 2n 2n. हर एक की 2 क्रोमैटिड कॉपीज प्रिपेयर कर लो, फिर वापस उन कॉपीज को एक एक में सेपरेट कर दो. So each daughter cell will be same like that of what? Mother cell. That is how you have to understand it. Cell entering in its cell cycle with 2n number in the G1 stage. During the S stage beginning, it is still 2n. By the end of S stage, it goes to 4n number. Same will be maintained from beginning to end in the G2 stage. And now at the end of the G2 stage with 4n number, the cell enters into division. And there, passing through M phase, it gives rise to two daughter cells. Both are again receiving 2n number of chromosomes in them. This is the representation of the entire cell division. Now, if you remember, which two activities have been included in the cell division? M phase is further broken down into which two phases you remember? Rudra, horribly wrong. Interphase is a horribly wrong. Karyokinesis and cytokinesis. Heth and Galav have replied correctly. M phase is further divisible into karyokinesis and cytokinesis. What is the meaning of karyokinesis? Nuclear division, division of nucleus, yes. If you see carefully this nuclear division itself, karyokinesis itself is further divided into subphases such as prophase, metaphase, anaphase, and telophase. Pro is related with the beginning. Meta का meaning होता है after. Almost similar is that in ana. Telo का meaning होता है end. Pro is for beginning or initial part. Telo is for end. So prophase, metaphase, anaphase, and telophase. 4 subphases in which karyokinesis is accomplished, and then cytokinesis also. And with that, mitosis division is over. Is this clear on your part? Then tell me, which are the 4 subphases in a right sequence to start with the first till the last? Which are the first one? Prophase in karyokinesis. First is prophase. Next to that, metaphase. Next to that, anaphase. Next to that, telophase. Yes, that is correct. So let's go in detail of these 4 phases to understand the mitosis. Mind you, one thing before I go in detail. In the mitotic cell division, when it progresses, it is a continuous process. It actually doesn't go in this way that prophase completed, then there is a gap, time gap, and then metaphase, again its completion, and then there is a pause, and again anaphase. Nothing like that. What happens is that it's a continuous process, starting to end, mother cell to daughter cells, karyokinesis, and later cytokinesis. Point is that for our understanding of this whole process, we have divided it into 3, or rather these 4 phases, that is prophase, metaphase, anaphase, and telophase. So that is our understanding about these phases. And you see each phase is marked by specific features. Specific features will define each phase. So that is how, looking at those characteristics in the cell, you can say that either the cell is passing through prophase, or it is passing through metaphase, or it is passing through anaphase or telophase. So when you observe any cell, you can make out whether it is in interphase or in division phase. Even if it is in division, you can make out whether it is passing through prophase or metaphase or anaphase or telophase. Everything is based on appearance of the cell, its cytoplasm and its nuclear content. You can make out chromosomal content, particularly you can make out which stage it is passing through. So we shall be learning about this mitosis in detail, but only after a break of 5 minutes, and then straight away we will begin with prophase. Break for 5 minutes. Stop the recording, ma'am. Fine. So let's talk about prophase. To understand prophase, first of all, we must understand what is the status of the cell in interphase. Let's assume this cell is entering into the prophase. So this cell is in which stage, by the way? Which part of the interface this cell will be? Tell me. Exactly. End of its G2 stage. Having just completed its G2 phase, now this cell will enter into prophase, in which actually it is taking entry in the mitotic division, M phase. Could you understand what I said? Cell having completed this G2 stage of interphase is now going to enter in the cell division now, specifically in a prophase part of it. So before you understand about the prophase, let us understand about the cell which is in interphase and going to enter over here. Look at this stage. Cell membrane, nuclear membrane intact inside this inside this cytoplasm. The centrosomes, each with 2 centrioles, they are present over there. Then inside the nucleus, there is a chromatin material, the genetic material, and there is a nucleolus. That is about the features of a cell which is in an interphase. Now, what happens to it now? Now, if you remember, as soon as cell enters into the division, what happens to chromatin? The genetic material inside the nucleus is known as chromatin as far as it is interphase. The moment cell will enter from, condensation of chromatin will occur into chromosome. So moment the cell will enter from interphase to division phase, its genetic material will start condensation. So from the loose, indistinct thread-like structure called chromatin, these structures start getting condensed, get organized into structures which are known as chromosomes. Structures known as chromosomes, each with one centromere and two sister chromatids over there. You remember that? Each with one centromere and two sister chromatids. So that is what happens to the genetic material, number one. Number two, that was the first event. Huh? Number two, see, the moment the chromosomes start appearing in the cell, you realize that the prophase has started. The cell has started its mitotic division phase. Why? Because as long as the interphase is there, chromatin will remain in which status? Diffused status. So it will be very light in appearance. The moment the cell will enter in the prophase, condensation starts. As soon as condensation starts, the chromatin starts becoming thicker and thicker, so it starts appearing. Appearance of chromosome itself marks the beginning of division itself. It marks the prophase over there. If somebody asks you, what is the characteristic feature for the prophase? Say that appearance of chromosomes in the nucleus itself is a characteristic feature of prophase. It begins, it's a beginning, it marks the beginning of division, it marks the beginning of prophase over there. And each such chromosome, if you see clearly, then it will have two sister chromatids held together by the centromere. While this is happening inside the nucleus, inside the cytoplasm, the two centrosomes, each with two centrioles, they are trying to establish a mitotic spindle. What they are trying to establish is a mitotic spindle. What is a mitotic spindle? Let me explain. Before I go in detail of mitotic spindle, one thing you should understand that at this stage of, in the second stage, that is a metaphase of the mitosis, you can see chromosomes have to be arranged in a specific manner in the center of the cell. For that, a network of fibers is created. This network of fibers, it is known as mitotic spindle. It is known as mitotic spindle. And the one which will help in establishment of that are the two centrioles, one at this pole and another one at the other pole. Now I will explain it in detail. What is a mitotic spindle? See, for time being, if you forget about the nucleus and if you just focus upon the cytoplasm, you can see that there are set of two centrioles. Let's say these are the two centrioles. With the help of these two centrioles, with the orientation or guidance provided by these two centrioles, inside the cell from its one end to the other end, a network of fibers is getting established. This network of fibers is known as spindle. The name given to that is spindle because it is in a mitosis. This is happening. We call it as the mitotic spindle. We call this network as a mitotic spindle. You had a pair of centrioles. With their guidance, a network of tubules is created. This is called as mitotic spindle. इस नेटवर्क को क्या कहते हैं? माइटोटिक स्पिंडल कहते हैं. So first of all, are you all clear with that explanation that a network of tubules, obviously these tubules are microtubules. You have already learnt about them. Am I right or wrong? Chapter number 8, microtubules, their role. They help in cell division. So the network of microtubules established inside the cytoplasm of the cell to support the chromosome arrangement at the equator. अभी आपको पता था ना सेल डिवीजन टाइम पे मैंने आपको डायग्राम दिखाया था मेटाफेज का. Chromosomes arrange themselves and at the center of the cell, which is called equatorial plane of the cell. वहां पे chromosomes को arrange करना है ऐसे. And for that arrangement of chromosomes, to support the chromosomes at that position, you require a network of microtubules getting established in the cytoplasm. This network is known as mitotic spindle. These two centrioles which are available, the two centrosomes rather, because each having pair of centrioles, while they are heading, one heading towards this pole, another one heading towards the other pole, they gradually start establishing this network in between them. So that part, if you can understand, that as soon as prophase starts, these two centrioles start moving towards their respective poles. But in that process, they help in establishment of spindle, the mitotic spindle over there. Acha, this spindle is made up of microtubules. The spindle is made up of what? Microtubules over there. Like this. This spindle has three different types of microtubules in it. Let's see which are they. The small microtubules which remain around the centrioles at the pole, they are called as ester. They are called as ester microtubules. In this network of tubules, there are three different types of tubules that you need to understand. One of them, very short microtubules all around the centrioles at the pole, we call it as the astral microtubules. Then others which will develop from one pole to another pole, centriole at one pole to centriole at another pole. These ones are known as polar microtubules, or else we call them as the continuous microtubules because they are established from one pole to another pole. And then what about the third type of microtubules? Have a look. And these ones are known as chromosomal microtubules, or even you may call them as the discontinuous microtubules, or else you may call them as the kinetochore microtubules, because here there are kinetochores. I am sure you recall small discs around the centromere to which these microtubules bind. We call them as the kinetochores. So three different types of microtubules. Together, they establish what is called as mitotic spindle. Now, before I go to the next, if you have any question, you ask. If you have no question, then I'll switch over to the next part of the Prophase. No one has any question. No one wants to be wants this one to be repeated over there. Chalo, you are 45 plus students, approximately 45. I need 45 yes or clear, so that I understand that all of you have understood this topic. अभी तक सिर्फ 10 आंसर्स हुए, 10 रिप्लाईज हुए हैं. I want all of you to participate. I repeat, I have explained to you the assembly of mitotic spindle, the one in which a network of three different types of fibers, a microtubule fibers, is established in the cytoplasm that is going to support the arrangement of chromosomes at central region of the cell during the cell division. This includes microtubules, which are actually generated from cytoplasm through polymerization of tubulin. Each of this category, whether astral microtubules or polar microtubules or chromosomal microtubules, they all are born from the cytoplasm through the polymerization of tubulin. The centrioles at the poles just give them support at the end, support to which they will remain connected. Otherwise, they are not developed by the centriole or centrosomes at the poles. Remember, so these microtubules are originated from the cytoplasm through the polymerization of tubulin protein. So this is the way they are being developed and establishing themselves to form the mitotic spindle. The one category of this, that is the astral microtubules, are very short microtubules that tend to remain around only centrioles at the poles. We call them as the astral microtubules. The second one are polar microtubules, which start from one pole and end at another pole of the cell. So continuous one without any interruption, they are established. They are supportive one. They are called as polar microtubules or continuous microtubules. The third is chromosomal microtubules. They start from one pole and at the other pole, they are connected to the chromosome at its centromeric region, specifically kinetochore disc. Similarly, दूसरी साइड से एक आया ऐसा क्रोमोसोमल माइक्रोट्यूब्यूल जो उसी क्रोमोसोम को सेंट्रोमियर पे अटैच करेगा, साइड दूसरी साइड के काइनेटोकोर से अटैच करेगा. So such microtubules which originate from the poles and go and attach to the same chromosome, they originate in a pair. एक ही क्रोमोसोम की एक एक साइड पे अटैच रहते हैं. So they are the chromosomal microtubules or discontinuous, क्योंकि ये पोल से स्टार्ट होके क्रोमोसोम पे एंड कर जाते हैं. So they are called as chromosomal microtubules or kinetochore microtubules or continuous microtubules, like that. So this type of network is known as spindle, the mitotic spindle. Coming back to our original plan, by the way, ऐसा दिखेगा. Have a look. Check out. Now, while in a cytoplasm, the spindle is being formed gradually inside the nucleus. The chromatin has started, condensation has started turning into chromosome. The nuclear envelope starts disintegrating. आप चेकआउट करो इसको. 3 स्टेजेस में अगर आप इसको देखोगे, इंटैक्ट था यहाँ पे न्यूक्लियर मेम्ब्रेन, यहाँ पे डिसइंटीग्रेशन स्टार्ट हो गया. Majority of the part of that nuclear membrane has disintegrated. Can you see this back-to-back arrangement of the nuclear envelope gradually disintegrating? Is it apparent to you? Can you see that the nucleolus, which is there in nucleolus, has disappeared in this prophase? It gradually disappears over there. So disintegration of nuclear envelope, disappearance of nucleolus, you see. Also Golgi body, endoplasmic reticulum, these structures of the cytoplasm, they start disappearing in this manner. Clear or not? Clear or not? अच्छा, एक और point regarding the prophase, regarding the rather cell division, mitotic division in a plant cell and in animal cell. Regarding that, one important point to be discussed. Naturally, you can realize this thing. This is the plant cell, and here is which one? Animal cell. Mitotic spindle will develop with the guidance of orientation of centrosomes at the poles, as far as that is the case of animal cell. In case of plant cell, there also the spindle is created. There also the chromosomes are arranged on the spindle. But that is done without the centrosomes at the pole. I am sure you can recall animal cells possess centriole and centrosomes. Centrosomes are absent in plant cell. Do you remember? Yes or no? So mitotic spindle must be formed in both cases, whether plant cell or animal cell. But in animal cell, it is done with the help of pair of centrosomes, one each present at given pole. Whereas in a plant cell, the similar mitotic spindle is done without the centrosome. The one in which it is done with the help of centrosomes, it is called as amphiastral type of spindle formation. And the one in which centrosomes are not required, it is called as anastral type of spindle formation. दो type के spindle formation होते हैं. In animal cell, it is done with the help of pair of centrioles. It is called amphiastral. In plant cell, it is done without pair of centrosomes. That is called as anastral. How many have understood? देखो यहाँ पे क्या होता है, स्पिंडल फाइबर्स का एक एंड सेंट्रियोल से अटैच्ड रहता है, तो उसको सपोर्ट मिलता है. Because plant cells do not have centrioles, so that end remains attached to the cell wall. सपोर्ट ही चाहिए ना. प्लांट सेल में यहाँ पे कौन सपोर्ट देता है? सेल वॉल यहाँ पे. और एनिमल सेल में यहाँ पे किससे सपोर्ट मिलता है? सेंट्रियोल से. Requirement is support. The centrioles do not produce the tubules, they just provide support. तो अगर वो नहीं है तो that doesn't mean that spindle will not be formed. Spindle तो वैसे भी फॉर्म होने वाला है with or without centriole. Centriole है तो उसको सपोर्ट देगा centriole. अगर वो नहीं है तो सेल वॉल का सपोर्ट लेके ग्रो करेगा ये स्ट्रक्चर. Well, with that we conclude the prophase, the one in which chromatin has started condensation into chromosomes, so the material starts appearing. Each chromosome seems to be having one centromere holding two sister chromatids. Cytoplasm, the pair of centrosomes start heading towards their respective poles. In that process, they will help in establishment of mitotic spindle, a network of microtubules originating from the cytoplasm through the polymerization of tubulin. This network shall provide support to the chromosomes being arranged on the central plane of the cell. Astral microtubules restricted around centrioles, polar microtubules established from one pole to another one, whereas kinetochore microtubules or chromosomal microtubules from pole to chromosome at the center of the cell. Parallelly, the nuclear membrane gradually disintegrates. Early prophase में disintegration start होता है, late prophase तक वो complete हो जाता है. Endoplasmic reticulum, Golgi, they disappear gradually, disintegrate. Even nucleolus disappears. Plant cell will establish mitotic spindle without the involvement of centrosomes as they are absent. It is called an astral type of spindle formation. an animal cell will form the mitotic spindle with the orientation guidance provided by the centrosomes. That would be called as amphiastral type of spindle formation, which is the next stage after prophase in mitosis. या प्रो अगर बिफोर है तो मेटा का मीनिंग होता है आफ्टर. So before stage, after stage means prophase, metaphase. Let's talk about metaphase. It has been divided again as like prophase, early prophase and late prophase. This is also early metaphase and actual metaphase. See, this will gradually occur over few minutes to hours. So you can consider it into divided into early part and later part of the same stage. As we see that nuclear membrane has completely disintegrated. Chromosomal condensation is accomplished. You can see that chromosomes are the most condensed structures at this stage. As you know that G1, S and G2, the chromosomes remain in a chromatin state. So obviously they will be in a very diffused stage. Only in a mitosis, the cell division period is the one in which condensation will be there. So बाकी सब टाइम पे तो क्रोमैटिन होता है, क्रोमोसोम तो सिर्फ डिवीजन टाइम पे होता है. डिवीजन में भी प्रोफेज में कंडेंसेशन स्टार्ट होता है, तो इतने ज्यादा कंडेंस नहीं होंगे. मेटाफेज में कंडेंसेशन कंप्लीट होके क्रोमोसोम मोस्ट कंडेंस्ड वर्जन में अवेलेबल होते हैं, क्योंकि उसके बाद एनाफेज में डिवाइडेड हो जाएंगे और टेलोफेज में अगेन क्रोमोसोम में से क्रोमैटिन में कन्वर्ट होना स्टार्ट हो जाएंगे. So let's say that if I wish to observe the chromosome of any cell, which is the best possible stage in the entire cell cycle, which is the best possible stage in the entire cell cycle when one has to observe the chromosome, metaphase is the one. Early metaphase is the one in which all chromosomes are most distinct as the condensation is completed, each with one centromere and two sister chromatids. And what is present on the centromere sides over there, that material is known as what? That proteinaceous disc on either sides of centromere, to which this it will be known as what? क्या लिखा था? Yes, it is called kinetochore. Proteinaceous plates on either sides of centromere of each chromosome, the ones to which those chromosomal tubules will go and attach. That is called as kinetochore discs. So that is about the chromosome. I repeat, the maximum condensation of chromosome is completed. Maximum possible condensation of chromosome is done. So each chromosome is distinctly apparent. This is the best stage to observe the chromosomes of any cell. Each chromosome has two chromatids held together by the centromere, and this centromere on its either sides possesses proteinaceous discs, which are called as kinetochores. Now, as the nuclear envelope is lost, all the chromosomes are scattered in the साइटोप्लाज्म. एक इधर गिरा, एक इधर गिरा, एक इधर, ऐसे 46 है हमारे सेल में, तो ऐसे 46 क्रोमोसोम्स scattered हो जाएंगे within the cytoplasm because nuclear envelope is lost. So that boundary is over. Now no distinction between nucleus and cytoplasm. Everything is now cytoplasm and chromosomes are spreaded in the cytoplasm. Meanwhile, those Those continuous fibers तो must be establishing from one pole to another pole. But parallelly, those which are called as chromosomal fibers, they also develop. One from each pole. So parallelly, for each chromosome, two chromosomal tubules will develop, fibers will develop. One will be heading towards one side of chromosome, another one will be heading towards another side of chromosome, as you can see in the picture, and will go and bind to its centromeric region where kinetochore discs are there. To the kinetochore discs, these chromosomal tubules will attach. Once they are attached to each chromosome, as you can see like this, they are dragged towards the center of the cell. ये वाले को थोड़ा नीचे लाएंगे, जैसे इसको इधर लाएंगे, इसको थोड़ा ऊपर ले जाएंगे, इसको थोड़ा नीचे लाएंगे, इसको थोड़ा नीचे लाएंगे, सबको एक लाइन पे लाना है. These chromosomes will be arranged in the middle of the cell, and that is done by these chromosomal fibers over there. You see, polar fibers are from pole to pole, continuity, whereas chromosomal fibers are only from one pole to chromosome. How many could understand the same? This is the fully established mitotic spindle, which has arranged chromosomes on an equator of the cell. देखो, ये अगर अर्थ का जैसे इक्वेटर होता है ना, वैसा इमेजिनेशन करो तो ये सेल का क्या है, इक्वेटोरियल प्लेन है, at which the chromosomes have to be arranged. This is called congregation of chromosomes. Congregation of chromosomes at equator. At equator. Understood? Congregation of chromosomes at equator. This is arrangement of chromosomes at equator. See, इस पोर्शन को कहते हैं मेटाफेज प्लेट. ये मेटाफेज स्टेज में अपीयर होता है, इसको कहते हैं मेटाफेज प्लेट. ये अरेंजमेंट. The plane of alignment of chromosomes at metaphase is referred to as metaphase plate. ये पूरा एक आप इमेजिन करें कि एक सर्कल होगा जिसमें क्रोमोसोम्स अरेंज होंगे ना, सर्कुलर अरेंजमेंट होगी ना क्रोमोसोम की. सेल फ्लैट नहीं है, सेल एक स्फीयर है. Cell is a sphere. So in that spherical shaped cell, at the center of that sphere, chromosomes are arranged like this. All centromeres in one line. And then chromatids are hanging from that position. अगर ये ह्यूमन सेल होता तो ऐसे 46 chromosomes की अरेंजमेंट होती इक्वेटर पे. That arrangement is called as metaphase plate. Process is also known as congregation of chromosomes at equator. Do you understand this? Hi Anna. ये सेल में एक लाइन नहीं है, ये सेल में एक सर्कल होगा क्रोमोसोम का. ऐसे अरेंजमेंट होता है सर्कुलर मैनर में, नॉट इन अ वन रो. Well, that was about the metaphase. After the prophase comes the metaphase. The chromosomes have completed the condensation. This is the point when you can study the chromosomal morphology. Best stage to observe chromosome of any cell, each with one centromere holding two chromatids, sister chromatids. Each centromere has kinetochore discs, proteinaceous discs called kinetochore on its sides, the one to which the developing chromosomal tubules or fibers will go and attach and will drag this chromosome towards the center of the cell. As nuclear membrane is completely disintegrated, the chromosomes spread themselves get scattered into the cytoplasm. To this scattered chromosome, the developing chromosomal fibers will go and attach to the kinetochore sites of theirs, because some are in some part of the cell, others are in other part of the cell. They all are dragged towards the central region of the cell called equatorial position of the cell. Their gathering at the center of the cell is called congregation of the chromosomes at equator. And alignment of chromosomes is such that centromeres lie at the equator and chromatids are hanging freely. This arrangement is called metaphase plate. The plane of alignment of chromosomes at the metaphase plate is referred to as a metaphase plate. One information, at this stage, if somebody inside this cell in which before it enters in the cell division, in this cell, if somebody introduces a material called colchicine, so what happens is that inside this cell, chromosomal condensation to will occur. Nuclear membrane disintegration will also occur. Scattering of chromosomes in the cytoplasm will occur. So up to this stage, the cell will go from prophase to metaphase. But because of colchicine, which is inhibiting the polymerization of tubule, the spindle fibers will not be formed. Without them, the mitotic spindle will not be established. So you will have a cell in metaphase with fully condensed chromosomes scattered in cytoplasm, but no spindle. Without spindle, you cannot arrange them on equator, and without arranging them on equator, you can't go in the anaphase. This means you can arrest the cell at metaphase by application of a chemical called colchicine. If you have a culture medium in which you have kept some cells which are undergoing cell division regularly, and in that culture medium, if you introduce colchicine, a chemical that can stop cells tubulin polymerization process. So in that scenario, without microtubule, the cell might first reach to the prophase, complete the prophase, go to metaphase. But because microtubules are not formed, the mitotic spindle is not established, it can never cross over the metaphase. It will be arrested in metaphase, won't go in anaphase. सेल वहीं पे arrested रहेगा. Any question on the same? Any question regarding this? See, so far we have seen this much. Prophase and metaphase. Appearance of chromosome marking the prophase, arrangement of chromosome on an equatorial plate that is called as metaphase. Now next will be anaphase. Which lecture you have after this?
Sir, there is a,
Physics.
sir, there is a break till 12 o'clock. After that, physics.
Exactly. So I can go for this anaphase. Enough time. Now, prior to this, you remember that in metaphase, chromosomes were arranged on an equatorial plane like this. Array. Am I right if I show you that this is the way that metaphase was there? You understand that? Now, next, what happens is this thing, and that is called as anaphase. If this was meta, the arrow shows that next is which one? Anaphase. In anaphase, each chromosome splits. Each chromosome splits into two chromatids of it. आपको पता है कि each chromosome has two chromatids held by the centromere. So centromere of each chromosome splits and two chromatids get separated. One chromatid goes towards one pole, another chromatid goes towards another pole. This is a characteristic feature for what is called as anaphase. The migration of chromatid from the equator to pole, this chromatid will start migrating from equator and will be heading towards the pole. That is what happens in each chromosome. First of all, centromere will be the one, and later to that, following that is which one? Its chromatids that will be pulled over there. जब ये क्रोमैटिड पुल होता है टुवर्ड्स दी पोल, पहले ड्रैग होता है उसका सेंट्रोमियर और उसके बाद ड्रैग होता है उसका क्रोमैटिड. पहले ये सेंट्रोमियर पुल होता है, फिर उसके बाद उसका क्रोमैटिड खींचता है यहाँ पे टुवर्ड्स दिस डायरेक्शन. Do you guys understand what I am explaining? Yes or no? See, if you understand for each chromosome separately, individual chromosome separately rather, not each, individual chromosome, this is how it will appear. Let's say that this is the cell. What happens to each chromosome during this anaphase? You see, these are the two chromosomal fibers attached to the kinetochore of this centromeric region of this chromosome. Now they will be pulled in this manner. This chromosomal fibers over there, chromosomal tubules will be pulled in this manner. Because of this pull, the centromere gets splitted. It gets separated over there, centromere. And further these tubules are pulled. That will further separate these two centromeres and their chromatids towards their respective poles over there. Imagine this happens to all chromosomes. This happens to all chromosomes. Each chromosome has got two chromosomal fibers attached to it, one on each side. They will be pulled towards their respective pole. That will be responsible for cutting the centromere into two equal halves. Now, each centromere has its own chromatid, one sister chromatid with each centromere. And now this will be pulled towards the respective pole. So first of all, centromere is being pulled over there. Following to that, what is being pulled is the chromatid. And that's how the separation occurs. Even at this stage, these separating units are also known as chromosomes only. तो आप बोलोगे कि सर ये भी क्रोमोसोम और ये भी क्रोमोसोम, तो हाँ भाई, ये भी क्रोमोसोम, ये भी क्रोमोसोम। ये वाला जो क्रोमोसोम है वो टू सिस्टर क्रोमैटिड्स वाला क्रोमोसोम है। दिस क्रोमोसोम हैज टू सिस्टर क्रोमैटिड्स। इस वाले क्रोमोसोम के पास सिंगल सिस्टर क्रोमैटिड, बट वो है तो क्रोमोसोम ही। So at this stage you can even call them as chromosomes. ये अगर ह्यूमन सेल होता तो यहाँ पे 46 ऐसे क्रोमैटिड्स कलेक्ट होते और यहाँ पे 46 क्रोमैटिड्स कलेक्ट होते। That is what one has to understand. Same number of chromatids on each side are being pulled over there. 46 chromatids on one side and 46 chromatids on other side. They will be separated. ऐसे 46 chromosomes थे यहाँ पे, इसके एक एक क्रोमैटिड सेपरेट होके अब पुल हो रहे हैं। चलिए, एनाफेज में कोई डाउट है?
No, sir. Clear.
That pulling of centromeric region of each chromosome by the two chromosomal fibers of the opposite sides splits the centromere into two, each with one chromatid. This centromere and chromatid together is also known as chromosome. These two separated chromatids will be pulled, they will be migrating towards their respective pole, each with one centromere and one chromatid. That kind of a structure will be pulled towards its respective pole. पहले centromere drag होगा और उसके बाद chromatid drag होगा. The point which you have to understand over here is there is a proteinaceous complex named as APC, Anaphase promoting complex. It was this complex which allowed the cell to enter from metaphase to anaphase. If it is ensured that if it is ensured that towards the end of the metaphase, all chromosomes have been aligned at equatorial plane, then only cell is allowed to go for anaphase. If even one chromosome was separated, not aligned, APC will not permit the cell to move from metaphase to anaphase. That permission shall not be granted. How many have understood this? APC's role. towards the end of metaphase, APC, the set of proteins, check up the cell. If all chromosomes are properly aligned at equatorial plane, then and then only the cell will be permitted to move from metaphase to anaphase. Otherwise not. Anaphase promoting complex will promote the metaphase to anaphase transformation or transition only if all chromosomes are aligned. इसीलिए तो जब हम colchicine डालते हैं and chromosomal alignment is not there, then naturally APC shall not permit the cell's transition from metaphase to anaphase. Lastly, cell has gone from prophase to metaphase to anaphase. Now it has to move to last, that is telo end part of this division, which is called as telophase. Last is which one? Telophase. Have a look. Everything opposite to what was seen in prophase will happen over here. So for example, in this stage, chromosomes start decondensing into chromatin. देखो यहाँ पे decondensation हो जाएगा. जो प्रोफेज में chromatin was condensing into chromosome, telophase में chromosome will be decondensed into which one? Chromatin. That's the first thing. So individual chromosomes will not be seen now. Chromatin network will be seen soon. Acha, nuclear envelopes start assembling around the chromosomes that are going to be collected at each pole. I told you in anaphase, the separated chromatids will move from equator to pole. When cluster of these chromosomes get collected at each pole, the nuclear membrane starts reforming around them. So a nucleus comes into existence at each pole. Within one cell, there are two nuclei. These are called daughter nuclei. So two daughter nuclei come into existence at this stage. Apart from that, inside that, a nucleolus will start appearing, जो disappear हो गया था प्रोफेज में. फिर Golgi apparatus and ER will reappear, जो प्रोफेज में disappear हो रहे थे. फिर spindle will disappear gradually, disintegrate, जो प्रोफेज में फॉर्म हो रहा था. अगर आप प्रोफेज को याद करें, इसको exactly opposite कर दें. In prophase, in a cytoplasm, mitotic spindle was formed. In telophase, in cytoplasm, mitotic spindle is disintegrating. In prophase, chromatin was condensing to chromosome. In telophase, chromosomes are decondensing to chromatin network. In prophase, nuclear membrane was disintegrating. In this case, telophase, nuclear membrane is reintegrated around the chromosomes collected at each pole. In prophase, nucleolus disappearing, ER and Golgi disintegrating. In telophase, nucleolus reappearing, the Golgi and ER reappearing. And finally, at the end of telophase, almost towards this telophase itself, cytokinesis starts over there. जब telophase start हुआ तब से ही cytokinesis start हो जाता है. And finally, this divides the cell into two daughter cells. Two daughter cells come into existence at the end of this telophase. Once the chromosomes have collected at each pole at the end of anaphase. They start decondensation into chromatin. Meanwhile, they start being enveloped around by the developing nuclear membrane. Inside them, they start developing nucleolus also. Inside the cytoplasm, Golgi and ER start being formed. Mitotic spindle starts being disintegrating. All these events occur towards the end, that is the telophase. And with that telophase, finally, again, these are the two daughter cells joined now. Soon they will divide and will give rise to two daughter cells finally. One minute duration I give you in case if you have any question to ask. Well, here we have concluded the mitosis, significance of mitosis, checkpoints, and then introduction of meiosis that is included in our next lecture on Monday. Till then, I think Monday there might be something different. It might be a common lecture for offline, online. जो भी होगा देखा जाएगा. We'll see that. But that much we will try to complete on Monday. So with that, I conclude today's lecture. If somebody is having any doubt or question, can ask. Okay, lecture concluded. See you in the next lecture. Till then, goodbye.
Listen, student, listen.
Madam,
There will be,
the
yes,
recording first.
yes, sir.